Hello, good afternoon, or good morning, wherever you're watching this video. My name is Shane Dunn. This is a neurocommunication presentation for my Bio 515 class. So I'm going to post this on my YouTube channel. I show my students because this is a concept many students struggle with. So this could be a supplemental video that I will give my students sometime. I want to remind you guys that I am drawing on my computer. It is broken right now, so it's not touchscreen, so I'm drawing with a mouse. So my drawings will be terrible, but hopefully you can laugh at it and still learn. I normally do this type of information in two pictures, so for a rubric reason, I'm going to break it up into smaller, simpler concepts. But I still will draw two major pictures that will show everything I explain. Now, chemistry basics. So before I explain anything, let's explain some concepts real quick. So first, whoops, nope, there we go. This is a sodium ion, potassium ion, calcium ion, and chlorine ion. So if you don't know chemistry, that is sodium, potassium, calcium, and chloride. If you don't know what this positive is, that just means it is a positive ion. It's plus something. This right here is a negative ion. It's a negative something. Calcium is the only special one that's not 1, it's plus 2. I'll explain why is that important later on. I'm going to refer to these things not as ions because ion is a mouthful, but I'll refer to them as sodium, potassium, calcium, and chloride. Okay, the first concept that I want to talk about is graded potential. And what this basically is first off, we're talking about a nerve cell, which looks like this. Graded potential is mainly going to happen in the axon, which is going to be this guy right here. So a zoom up of the axon, as you'll see a bunch of positive and negatives. What I want you guys to know is that on the outside of a neuron, it's positive. On the inside, it's negative. And the reason for it being negative on the inside is that there's more amino acids and more proteins, which are always slightly chemically negative. So the inside's always negative, the outside's always positive. And the two ions we're going to focus on for the first part of this is potassium and sodium. Now, hint, hint, wink, wink. This question will always stump students and was very popular at Grand Canyon University. Potassium is always on the inside of the cell until something happens. The best way of knowing this is this little acronym I use, or metaphor, is king. Potassium is king on the inside. I like this because potassium starts with a K, and the word king has I-N in it. So if you write the word king down, you know potassium goes on the inside. And if potassium is the inside, sodium has to go on the outside. So there's all this potassium on the inside, all this sodium on the outside. But the inside is still negative because it has more amino acids and proteins. And now we're talking about two types of gates. So when you hear the word gates, you mainly think about something you open up. Most gates are locked. And how do you unlock a gate? Well, you use a key. Well, there are two types of gates we'll talk about, a voltage gated and a chemically gated. For graded potential, we're mainly going to focus on the voltage gated. Chemically gated is going to happen during the synapse. So a voltage gated gate opens up when a charge is met or when a voltage is met. So when a certain charge or voltage is required, the gate opens up and lets things go in and out. The next thing I want to mention is that potassium is relatively happy. Sodium is not so happy because, like a magnet, right, magnets have two ends, a positive end and a negative end. If you take two magnets that are, like, say, for example, has two positive ends, when they put the two positive ends together, right, they push each other away. They don't want to be with each other. They repel each other, right? Like you want opposites to attract. So a lot of sodium wants to go inside the cell, but it can't because there's no gates. There are gates though, and there are these voltage gated we'll mention a little bit. But a certain voltage has to be met, and that's what I'm going to talk about now. This drawing is something I want you to follow along. If you don't like the last picture, you can easily just do it in a couple of drawings. So here I'll draw all my potassiums. I'm going to draw 3, right? Sodiums are up here. Remember, I'm drawing with a mouse, so my drawings are not the best. Now I'm going to use, let's use orange. So this right here is going to be an open channel. And sometimes there are a few open channels that allow a drizzle in of sodium into the cell. And sometimes there is a drizzle allowing some potassium out. Very, very small though. This drizzle in and out helps maintain the resting potential, or the charge, right? Remember, it's always negative inside the cell and always positive outside the cell. Well, I told you when a charge is met, right? You have a gate. Oof. Right here is a gate. Here's a closed gate. The sodium can't enter in because it bounces off. If this gate were to open up, for example, sodium can come. Sodium can come rushing through. And what will happen when all the sodium comes rushing through? The inside becomes way more positive than the outside, and the symbols flip. So now the inside's all positive, and the outside's all negative. Only happens when all the sodium rushes in, right? Well, this is a good thing because this generates an impulse, or kind of like static electricity. When these things kind of rub against each other, it's going to create electricity, which allows our nerves to talk to each other. So this concept is going to explain action potential. And action potential is just how to generate the impulse within one nerve cell. Here are the concepts. I'll explain it in the next drawing. Negative 70 millivolts, that is the resting membrane potential. Negative 55 is threshold. Now, when I hear the word threshold, I want you guys to kind of think about a gun. Most of you have shot a gun before or know what a gun is, but a gun is fired when you pull a trigger. Now, don't do this because guns aren't toys, but if you just tap a trigger without putting a lot of force on it, it won't fire. But as soon as you put enough force on a trigger, it's going to pull back, the hammer's going to cock, and the hammer's going to hit, the bullet's going to fire, right? What threshold is threshold is putting enough pressure on the trigger to cause the gun to fire. So basically, when you think of threshold, think of a gun firing. The trigger is pulled, the gun will fire. There is no stopping it. If enough pressure is gone or enough voltage is met, action potential will go off and impulse is generated. Negative 55 to positive 30 is something called depolarization. D means without or to stop polarization. Re means to reapply. Polarization, polar just means poles, a positive pole and a negative pole. So polarization is when the poles are going to flip. I kind of showed you the last one. So depolarization is when all the negative flips out to the positive and all the positive flips out to the negative. Repolarization is the exact opposite. And we have these two periods here, an absolute refractory period and a relative refractory period. Absolute means it, without a doubt, nothing can happen. Relative is, well, relatively if this or that occurs, this could happen, and I'll explain going on. So this is a normal action potential graph of a neuron cell. Number one is going to state the resting potential, negative 70 right here. It would stay negative 70 as long as nothing happens, but you move your body, you think, so things do happen. So what happens is a nerve will receive signal and let sodium into the cell. This will happen, and we'll explain where this occurs later on. But sodium will enter the neuron membrane, and this actual neuron rushing in, right, all these positives will bring the negative 70, negative 55. And like pulling a trigger of the gun, as soon as you hit negative 55, boom, you fire. It goes all the way up. 2 is depolarization, right? So what happens with depolarization, remember I mentioned those voltage gated? Well, negative 55 is the voltage gate for sodium channels. So now all the sodium channels that were outside the cell now rush in. All that sodium rushes in makes the cell way more positive, goes all the way up to positive 30. That's this right here. This section right here is depolarization. Number 3 is going to be repolarization. Well, number 3 is mainly going to be where repolarization can occur. But I'm going to say that's 4. 3 on this hand, though, because these graphs are different depending on who you teach. But what happened with 3 on this graph, what it's trying to tell you, is that the voltage gated for potassium channels open up, right? Well, potassium and sodium are all inside here. They don't want to hang out with each other because now all the positives are inside. So potassium's going to leave. They're going to mass exodus of potassium. Well, all the positives are leaving now, and it's going to bring it back down. 4 is repolarization. And if you notice, it dips way down here to negative 90. This is hyperpolarization. Hyper just means a lot. So it gets all the way down to negative 90, but then it slowly goes back up. So I'll show you in color. 2 is going to be depolarization. 4 is going to be repolarization. And 5 is going to be hyperpolarization. Okay. So all this happened within seconds. So this is, and MV right here is just membrane potential in millivolts. So we're talking about 1/millionths of a voltage. These are very, very, very small numbers. This happens instantaneously. But this, every time a neuron wants to impulse, this process occurs. Now, let's talk about a couple things. I'm going to use, let's use orange. So relatively, well, actually, the absolute period is right around here. So anything above this orange line, it's usually shown like this, is your absolute refractory period. And what this means is, if the neuron is in this moment in time, it absolutely cannot fire again because it's already fired, right? When you shot a gun before, you have to pull the trigger back, the hammer's got to hit the primer's got to fire, ignite the gunpowder, the bullet leaves, right? You can't shoot another bullet until the bullet leaves the chamber. Well, this moment right here, the gun or the neuron's still technically firing, so you can't fire again, you have to wait. As soon as you pass this orange line, you enter the relative, right? So maybe I can fire. I'll show you I'll show you right here with this blue right here. All this is relative refractory period. I can fire if my other neurons tell me to fire enough. We'll explain that a little bit. This is all the relative. Now let's talk about real quick how we get from 5, this is negative 90, all back to negative 70. This is going to happen with the sodium potassium pump, right? Oh no, all my sodiums are now on the inside, all my potassiums are on the outside. What's going to happen? Well, I got to pump them, all right? So you got to go against this, with or against the concentration. So this process is active. This is an active process, which means it requires energy. The energy is in the form of ATP, right? Because all the sodium are happy. All the sodium want to be inside the cell, right? All the potassium want to be outside the cell. They don't want to be all near each other. But we're forcing them to be unhappy. Because when you force the sodium and potassium to be kind of unhappy, they rush in with such vigor that an action potential is caused. Electricity they rub against each other. Electricity is formed and impulse happens. We spend energy to do this, and it's for every 3 sodium, 2 potassium. Let's actually use this in red. Sorry. For every 3 sodium we push out, we bring 2 potassium in. The best way I like doing this is the year is 2021, right? So I put 2K21. This 2K means 2000, but I also know it as if 2 potassiums come in, right? 3 sodiums have to leave. As long as you know one of the number, you should know the other. So it's going against the concentration, and it is an active process. It requires energy. Cool. Now let's talk about the structures of a nerve cell, because this is going to help us do our impulse. So some structures include our dendrites, which is the beginning, our soma, our axon, our terminal branches, and what I like to make a big deal about is right here is the axon hillock. This is where all active potential is generated, is the hillock. Okay. So structure we mentioned voltage gated, now we're talking about chemically gated. Vesicles are a little bit fat bubble, we'll see that, neurotransmitter and receptor. So here's what this is what the terminal at the very, very end. So let's actually go back real quick. Oops. Sorry. We're talking about this stuff right here. We're looking at these synaptic terminals. So as the active potential goes on, it sends electricity along the edge, right? These will allow these voltage-gated calcium channels to open up. Calcium will rush in, right? And since calcium is +2 charge, not a lot is required to rush in. And when it rushes in, it meets these vesicles or these fat bubbles. And these fat bubbles don't want to deal with all these positive charge, so they're forced to leave. And inside of them are neurotransmitters. These neurotransmitters are going to fling out to another dendrite, another neuron, right? The dendrite receiving side, and bump into receptors. This will allow a chemically gated channel to open up, and it'll either allow sodium to enter in, or it can allow chlorine to enter in. So when sodium is let in, the cell's going to fire, but if chlorine is let in, remember we're starting at -70. If chlorine is let in, it's going to go down to -75, maybe all the way down to -90. And it makes it harder and harder to fire. So the function of a synapse, and why it's very important to know, is because they send signals to each other. If I wanted to move my mouth when I'm talking, right, my brain's got to talk to my lips. And how does it do that? Well, it uses synapses, and they're going to talk to each other. All your nerves are not really connected. They're a little slight difference apart, but it's not one single nerve. It's a bunch of functions. So we have inhibitory postsynaptic potential and excitatory postsynaptic potential. Now, when I mentioned that up there, I mentioned chlorine, right? Chlorine is a negative one. So when chlorine enters the cell, right, it's going to go from -70 around -90. This is inhibiting the cell. Oh no, it's inhibiting the cell. Inhibitory means to inhibit or stop. Postsynaptic, post means after. So it's going to be fired, it's going to be allowed in, and it's going to affect the cell. Excitatory is a go, go, go. This will be, for example, like sodium. And we'll see in some of the muscles, it'll be sometimes calcium's rushed in. So let's draw it out. All right, let's draw out, here's a terminal branch, here's another terminal branch, and here's one right here. So an active potential is generated, electricity goes down, calcium rushes in. When calcium rushes in, it meets the vesicles, bop, 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 bop. These vesicles leave, and in this case over here, I'll make these vesicles contain chlorine over here. Well, technically they don't contain chlorine, they contain neurotransmitters that allow chlorine to go into. Same thing over here, vesicles, calcium comes in, da da, da da da, these leave, and sodium will come in. If I have more sodium than chlorine, this guy right here will fire because he has got receptors that look like kind of like hands that grab him. Cool. Okay. Hope this video was helpful. Here's reference slides I did. Hopefully all was helpful. Thank you. Have a great day.